Hi, Assalamualaikum. Thank you for joining. My name is Azza and I'll be sharing some interesting findings from my systematic review of effects of polyphenols in tea on modulation of gut microbiota in human trials and animal studies. These are the contents in this video. Tea has been known for the longest time as the second most consumed beverages in the world next to water. Because tea is less absorbed in small intestine, they end up in large intestine and becomes bacterial fermentation substrates. In there, microbiota will increase the tea bioavailability, which in turn, tea will suppress or promote the bacterial growth. Therefore, this review will gather all evidence on the effects of tea intakes on modulation of gut microbiota composition in order to know the effective types, forms, dose, and duration of tea intakes to have such an effect, since there are no previous systematic review were done. The eligibility criteria for the articles include English randomized control trials and animal studies from the past 10 years. The human subjects should be male and female with normal and overweight BMI, non-smokers, non-alcoholic, free from medications, and have low polyphenol diet. They must be given tea supplementation as case and placebo or no treatment as controls. Outcome measure is diversity analysis, which includes alpha diversity, which is the within sample diversity. That includes estimators such as richness and evenness, and relative abundance. The second one is beta diversity, which means between sample diversity. Articles were searched in PubMed and Scopus database using these keywords. Initially, my original proposal was to look into human trials only, but the articles are too limited, so I have to spend more time and research to look further in animal studies. Results analysis. Prisma flowchart show how the articles are selected. Through 3008 papers, only 21 papers are included in the review. 6 are human studies and 15 are animals. Findings in human trials. In terms of study design, 3 papers are RCT, 3 is pre-post intervention. Study duration is between 10 days to 1 year, and these trial subjects are quite diverse. 2 papers on healthy subjects, 1 hypercholesterolemic, 1 postmenopausal women, 1 non-habitual tea drinker, and 1 Caucasian population. According to Jadad score, 2 papers are strong, 1 moderate, and 3 low. All the studies use different kinds of tea with different food metrics and also different dosage, except for black tea who has no data on their dose. In terms of richness, only 1 study noted increase in beneficial bacteria called Bifidobacteria species. As for richness and evenness, only 1 study noted increasing trend. As for beta diversity between treatment and non-treatment group, only 1 study noted clear separation of the bacterial profile, which is a sign of modulation. Looking at the dominant phyla of the bacteria, there are mixed results between 2 studies. Bacteroidetes is decreased in 1 study but no changes in another study. Firmicutes is increased in 1 study but no changes observed in another study. No changes in actinobacteria. While the other papers reported bacteria in terms of their functions, but none of them reported the same thing. Although there is only small studies, each of them has different duration, quantification methods, types of tea and dosage, and food metrics. Therefore, no solid conclusion can be made due to mixed results and high inconsistency in the studies. It just reflects how unique and stable our microbiota is. In animal studies, all were done on mice, with the duration from 30 days to 26 weeks. The subjects used are ranging from healthy rats, diabetic, human flora associated, obese, and UVB exposed. And we can see a pattern here where almost all are given high-fat diet. For tea, they were given either tea extracts or infusion of the unfermented version of tea, which is green tea, black tea, oolong tea, or the fermented version, which is pu'er tea or fuzwan tea. The dosage used is different. When we compare, those with high-fat diet has reduced richness, but when the diet is combined with tea, the effects are totally reversed. And the same effects are seen in richness and evenness estimators as well. In most obesity cases, reduced bacteroidetes, increased firmicutes, and increased ratio of firmicutes to bacteroidetes has been linked as obesity markers. And these trends are seen in mice with high-fat diet, but addition of tea has reversed the trend. For beta diversity, all animal studies showed clear separation between the treatment groups and non-treatment groups. The limitation is, these papers use different dosage although they use the same types of tea. In conclusion, tea potentially modulates the gut microbiota through rebalancing the changes occurred. Interestingly, the effects are seen in all tea types, dose, and duration. Anyhow, usage of animal equivalent dose formula is needed if we were to interpret them into human. These are the references used. And these are the links for evidence table. Thank you for listening.